Hello, welcome to demola, and today's lesson we are covering enzymes. Really nice topic in terms of gets you thinking about previous ideas we've had, and also very important in terms of biological reactions in the body. So we're gonna get straight into it.
Okay, so whenever we're talking about enzymes, we need to do definitions, right? And an enzyme, simple biological catalyst. Yeah, it increases the rate of chemical reactions in the body by lowering the activation energy without being used up. Yeah, just remember that, please. And we talked about globular and fibrous proteins in the last lesson. Enzymes are globular, right? So they have a specifically shaped active site because of the specific bonding. We're going to get into that in a moment. But globular proteins dissolve in water, 3D structure, tend to be spherical. Yeah. See this diagram here. The main thing we really want to talk about is the active site. Yeah. Really important because that's where your substrate's going to bind. So these always have a very specific shape. We are going to talk about how it gets that in a moment. Yeah. But that's what's going to bind. Please, I will say it right now. Do not say binding site, say active site whenever you're talking about enzymes. Cool. Extracellular versus intracellular, I'm not too fussed with that. This just looks like a U, so we'll change that. But not too fussed. Extracellular works outside the cells, intracellular works in the cells. So, like, for example, maybe something like a lipase is going to work in the bloodstream, for example. And intracellular, I can't think of one off the top of my head right now. But even something like DNA polymerase is an enzyme. Works inside the cell. Cool. What I really want to get to is exam question: how do enzymes have a specifically shaped active site? This will come up every single year without fail, so it's really good to know. But what you want to say is a gene has a specific sequence of DNA, transcribed to produce a specific sequence of codons, in mRNA, translated to produce a specific sequence of amino acids joined by peptide bonds, or primary structure. R groups are in specific positions, so the bonding between R groups, ionic bonds are sort of bridges. Always say an example. Whenever you talk about bonding between R groups, just say an example. Examiners love it. Yeah. And then causes the protein to form a specific 3D structure with a specifically shaped active site that is complementary to the substrate. Now, you want to love these words. Yeah. These words, love them, please. You want to love the word complementary. And you can see how many times I mentioned specific. It's like, oh my goodness. It almost gets a bit annoying how much you need to say specific. But they love seeing specific. Yeah. Just say it every single line. And you can see here, we're kind of just regurgitating in a simplified way protein synthesis. So if you know protein synthesis as well, that's going to help you a lot of answering questions on this topic. Yeah. That is the spiel, the answer that you want to say every single time. So in terms of how enzymes do their thing, there's two theories, right? First one you would have known from GCSE, lock and key hypothesis. It's that the enzyme has a specifically shaped active site that is complementary to the substrate, right? Substrate just fits on in there perfectly. You form what is called an enzyme substrate complex, really important. And then after that, you put a strain on the bonds between the R groups. Again, for example, ionic bonds, and then that substrate is going to split off. Yeah, simplified diagram. What's new is the induced fit. Yeah, I've exaggerated it here. It's not going to have all this space there, but in the induced fit model, it's that your substrate doesn't fit perfectly into the active site, right? What's going to happen is the substrate's going to come on, and then your active site is going to kind of mold around it. Yeah, that's that molds around it. That's going to put a stronger strain on the bonds because you're really going to push at them, right? Causing them to break. Same thing. Yeah. Why maybe do we use induced fit over lock and key? Well, can you imagine? Say if this guy isn't coming perfectly head on. Yeah, he's not coming perfectly head on. Maybe it's the same substrate, but maybe he's coming at an angle. That one might not fit into that site. Yeah, this is gonna have to contort itself to try and fit in. Whereas in this model, if we're coming at a bit of an angle, we're still gonna get in, and then we can just mold the active side around it. Yeah, so the active sides are still complementary, right? They just aren't exactly the perfect fit yet to kind of allow a bit of leeway. And we're also going to want to discuss factors affecting the rate of enzyme-controlled reactions. So there's four main ones that we want to cover. A lot of them are quite similar. So temperature and pH, yeah. You should know this from GCSE. Let's focus on temperature first. You increase temperature, you're going to get to what we call an optimum, yeah. At the optimum, this is when your enzyme's like, I'm in my zone, yeah. I'm in my zone. I'm going to increase the rate of reaction as fast as I possibly can, yeah. When you go above the optimum for temperature, when you go above the optimum, then we have problems, yeah? Because then you're going to start breaking bonds between R groups. For example, best one I have is hydrogen bonds, yeah? break the hydrogen bonds, meaning that your protein is going to unfold. Yeah, and when it unfolds, there's a change in the shape of the active site. Yeah, and when you have a change in that shape of the active site, this is called denaturation. Yeah, you denature the enzyme because the active site changes. It's no longer specific or complementary to your substrate. So we cannot have this reaction keep on going at a fast rate. Similarly with pH, it's kind of the same thing about denaturing. I would maybe say instead I'd say ionic bonds instead of hydrogen bonds if you're going for the example. And it's just that, like with pH, for example, we have one where pH is like fairly neutral, it's optimum pH, but you can also have it like maybe the graph looks like this, where the optimum pH is more acidic, and same kind of way where you can have it on the other side where the optimum pH is alkaline. Now we need to talk about enzyme substrate concentration. This is a graph that I think is just good to know. Like, what you really have happening here is that active sites are going to be occupied. Yeah, if you keep increasing the enzyme concentration, you get to a point where you can no longer fit. So there's not enough substrate to fit all the active sites. So that's why we can't have an increase in reaction when we increase enzyme concentration. And similarly with substrate, that gets to a point where all of the active sites are occupied, and that is your point of saturation. And then you can't increase the rate of reaction by increasing substrate concentration. Okay, but there are also going to be times when we need to inhibit an enzyme. Yeah, why might we want to inhibit an enzyme? Maybe it's doing too much. Yeah, enzyme's too overactive. Or maybe it belongs to a harmful microorganism, a bacteria, or a virus. Other thing could be that it's making a harmful product, yeah? Either way, we just want to say enzyme, cut it out, yeah? Cool. We can do this in two ways. Competitive inhibition, yeah? A competitive inhibitor has a shape that is specific and complementary to the active site, otherwise identical to the substrate, yeah? So it's also going to be able to occupy the active site. And in some cases, it is better than doing that for the substrate. Or maybe you've just got a higher concentration of the competitive inhibitor, so it's occupying more of the active sites compared to the substrate. Yeah. When that happens, the substrate cannot bind. Yeah. The binding site's gone. It's gone. It's taking up the space, so this guy can't bind. Yeah. You've inhibited the enzyme's activity because it's not going to be able to break down the competitive inhibitor, but it's also not going to be able to bind to the substrate. What about non-competitive inhibition? Well, there are special things that we call allosteric sites. Yeah, so this is an allosteric site. I'm going to highlight the word here. Yeah, your non-competitive inhibitor is going to target this allosteric site. Yeah, goes to bind onto this allosteric site, and that's going to mean that the active site changes shape. So you can see here, this active site was initially going to be like a rectangle shape. Now it's a triangle shape. It's not going to fit in a substrate anymore. It doesn't fit the substrate, and that is how we can do non-competitive inhibition. Yeah. So two different methods. One of them, we're going to directly go into the active site, make sure the substrate can't get it. The other way, we're going to change the active site. Substrate still can't get him. Okay, so a couple questions to try. We got the first one, explain the importance of a primary structure for an enzyme. Next one, calculate your Q10. If you need to look up how to do that, you can find it online. And then just a question about how temperature affects the rate of reaction. So those are 3 questions. Have a Okay, so here are your answers. So for the first one, we're talking about primary structure and how it affects the reaction. Now, and how it affects the active site. Cool. When I'm answering this question, I'm going to go on a spiel that initially doesn't get me marks, but it does enough of the groundwork that I am going to get marks, regardless of what the mark scheme says. Yeah? What I start with is that primary structure is a sequence of amino acids joined with peptide bonds. Sometimes you get a mark for that. In this case, you don't. But sequence of amino acids joined with peptide bonds. Yeah? Specific sequence of amino acids. Cool. So I'm going to then say specific position of R groups. Yeah? I'm literally following that order. I haven't gained the mark yet, but in other questions I might have. Yeah? Determines the interaction between R groups, bonding between R groups. And again, I'd name an example, like ionic bond, the sulfide bridge. But you can see here, you finally gained the mark. Yeah? We're going to keep going. If the bonding between R groups happens in specific positions, we fall into a specific 3D shape. That's our second mark. Then, I'm going to say, specific 3D shape means that we have a specifically shaped active site, which is complementary to the substrate, which is ATP. Yeah? And you can see here, effective state of the active site, complementary to ATP. So you can see here, we've gone on that spiel. Again, maybe the first bit we didn't get any marks for this question, but in different questions we would have. Yeah? We have to tailor it to whatever they put in that mark scheme, I know I'm going to gain marks. Cool. Next, Q10. Q10 is like, it's a calculation that you do to see by what factor the rate of reaction increases when you increase your temperature by 10. So if you go back to this, you can see between 20 and 30, there's a difference of 10 degrees. Yeah. You just do 240 divided by 80. Yeah, so higher temperature divided by lower temperature, that will get you your Q10 of 3. Cool. So this is your answer here. And then finally, effective temperature. If you're a chemist, you're going to love this question. Yeah. More kinetic energy. Yeah. If we have more kinetic energy, more frequency of collisions. Yeah. And then this is when we get into maybe a bit of biology. More enzyme substrate complexes form. That on itself, I think is good enough. Yeah. If you were being absolutely greedy, then you can mention out the Q10 we just worked out. So the activity triples. And to be honest, it is a bit greedy, but I would say it anyway. Like, you just worked out that the activity tripled. You might as well just throw it in. Yeah? That's how you go about answering these. Okay, so really with the A-star tips, not too much for enzymes, but really important, specific and complementary. They need to become your best buddies, yeah? Every single time, specific and complementary. Cool. Protein synthesis. You saw when we were talking about the question at the start, that question comes up almost year on year without fail. Yeah. So knowing protein synthesis is going to be really important so that you can actually describe that process. Other things, ketones, activation energy, active site catalyst. Just make sure that you know how to define them. Yeah. Again, you could argue that at least 1 and 3 are more chemistry terms. But if you're not able to define them, you're going to be chilling. Cool. What do we not do? Binding site. This is the one that really gets a lot of people. If you say binding site, they are not going to give you the mark. Yeah. Make sure to say active site. Yeah. Because binding site will not get you the mark. And then again, the graphs for the factors, just be aware of them. I don't think they come up too often, but just like knowing that that's going to happen is really important. Yeah. That's going to be a wrap for the video today. So I hope that was really helpful. If it was, please like and subscribe to the channel. Any questions at all? I think I've been in my bag with replying to comments, I hope. So if you have any questions about anything, you should drop it in the comments below. I'm going to do my best to answer in a timely manner. We've got the TikTok and the Instagram, both at demola. But otherwise, that is it. So I hope you enjoyed the video. Please again, like and subscribe, and I will see you in the next one. Bye for now.